Hi, I'm Pipa Empai, a medical student from West Bengal. This is the very first video of All About ECG series. So, whether you are a medical student, a healthcare professional, or just someone eager to understand how the heart's electrical system works, this series is for you. So, without any further delay, let's start. What is ECG? Graphical presentation of electrical activity generated by the heart is known as ECG. That means if we graphically represent the electrical activity of heart, this graphical representation is known as ECG or electrocardiography. So what are the uses of ECG? Number one, diagnosis of heart disease like arrhythmias, myocardial infarction, coronary artery disease, and cardiomyopathy. Monitoring heart health like hypertension related changes, effect of medications, post surgery, or pacemaker monitoring can be done with the help of ECG. Number 3, emergency situations. Cardiac arrest detection, electrolyte imbalance, pulmonary embolism can be diagnosed with the help of ECG. And ECG also used in preoperative and routine screening. Now, to understand ECG, we must know few points about the ion channels, ion concentration, inside and outside the myocardial cell, resting moment potential, threshold potential, and few points about the heart. So let's start one by one. Number 1, ion channels. There are 3 ion channels we must know to understand the ECG. Number 1, voltage gated sodium channel. Number 2, potassium channels. And number 3, calcium channels. Okay, so these ion channels we must know to understand the ECG. Now, let's discuss about intracellular and extracellular concentration of sodium, potassium, and calcium. So, ion concentration. So we must know about the 3 ions. These are sodium ions, potassium ions, and calcium ions. So intracellular and extracellular concentration of this ion are important. So intracellular and extracellular. So intercellular concentration of sodium is about 12 milliequivalent per liter, and extracellular concentration is about 140 milliequivalent per liter. For calcium, it is about 140 to 150 milliequivalent per liter, and extracellular is about 3.5 to 5 milliequivalent per liter. In case of calcium, the extracellular concentration is 1 to 1.4. The unit is important here. It is not milliequivalent, it is millimole per liter. And the intracellular concentration of calcium is 1000 times less than the extracellular concentration. Okay, so you can remember those values. It's okay if you can't remember this, but you must know that the extracellular concentration of sodium is more, that is about 140. That means extracellular concentration of sodium is more and intracellular concentration of calcium is more, so these are opposite. Okay, so you have to remember that the extracellular concentration of sodium is more and extracellular concentration of potassium is less. Now we are going to discuss about the threshold potential. So let's discuss about threshold potential. I am taking help of a pie card of threshold potential. So what is threshold potential? Threshold potential is the minimum level of depolarization that causes an action potential to occur. Okay, so what is threshold potential? Threshold potential is the minimum level of depolarization that causes an action potential to occur. That means if a cell reaches its threshold potential, it can exhibit action potential. So it is the membrane potential at which voltage gated sodium and calcium channels are open. Okay, so let's draw a cell membrane and see the threshold potential. So that is the lipid bilayer of cell. I am drawing. So that is the cell membrane. Now we know inside the cell there is negative charge and outside the cell there is positive charge. So this potential, where there is separation of positive and negative charge, so here is a potential developed. So this potential at which the cell can generate an action potential is known as threshold potential. Okay, so we are discuss about the ion channels, the ion concentration, and number 4, the threshold potential. Now let's discuss about the resting membrane potential. So let's take another pie chart to understand resting membrane potential. So that is the pie chart of resting membrane potential. So it is the electrical baseline of cell. That means resting membrane potential RMP. It is the electrical baseline of cells. The definition is like that: the electrical potential difference across the cell's plasma membrane when it is not active or stimulated. So this point is important. It is the potential difference across the cell membrane when the cell is not stimulated. Okay, now resting membrane potential is maintained by sodium potassium ATPase pump and the large negative proteins inside the cells. So let's draw a cell membrane and sodium potassium ATPase pump. So that is, as usual, lipid bilayer, that means cell membrane, and here is a sodium potassium ATPase pump. Let's suppose that is a sodium potassium ATPase pump. Okay, so this is an active pump. Okay, that means it uses ATP and converted it to ADP, and with this energy two potassium ions get in and 3 sodium ions loses from the cell. So 2 potassium gets in and 2 sodium gets out. So with the mnemonic of Nokia, you can remember this, NOKIA, that means sodium out, potassium in. So 3 sodium out and 2 potassium in. So net charge will be positive on the extracellular part and negative in the intracellular part. And to aid this negative charge, here are larger negatively charged proteins. These are negatively charged protein inside the cell. So that is the resting membrane potential. So what is the definition? The electrical potential difference across the cell's plasma membrane when it is not active or stimulated. So let's discuss a few values of resting membrane potential of different cells. Okay. So resting membrane potential of skeletal muscle cells is about minus 95 millivolt. In case of ventricular myocytes, it is about 90 millivolt. In case of atrial myocyte, it is about minus 80 millivolt. And in case of AV node, it is about minus 60 millivolt. And resting membrane potential of SA nodal cell, let me know in the comment section. So you have to comment the resting membrane potential of SA nodal cells. Okay, comment below. So we have understood the ion channels, intracellular and extracellular concentration of ions, threshold potential and resting membrane potential. Now let's discuss few points about the heart. So our heart is composed of 3 types of cardiac muscles. Okay, number 5 about heart. It is composed of three types of cells, 3 types of muscles. Number 1, atrial muscles. Number 2, ventricular muscles. And number 3, specialized excitatory and conductive muscle fibers. So in short, specialized cardiac muscles. So specialized cardiac muscles like SA node, AV node, the Purkinje fibers, these are under the specialized cardiac muscles. Now the atrial and ventricular muscle contract much same way as skeletal muscle. That means contraction of atrial and ventricular muscle and the skeletal muscle are almost same, but the main difference is the duration of contraction. Okay, duration of contraction. Duration of contraction of skeletal muscle is less, but in case of cardiac muscle it is more. That means duration of contraction of cardiac muscle is more than that of skeletal muscle. Okay, now let's see the cardiac muscle cells. So I have already drawn it in a pie card. So that is cardiac muscle cells look like. Okay, so this dark area crossing these cardiac muscles are called as intercalated discs. Okay, I think you have already heard about the intercalated discs. So they are actually cell membranes that separate the individual cardiac muscle cells from one another. That means this is a cardiac muscle cell and this is a cardiac muscle cell. These are separated by this intercalated discs. So at each intercalated disc, the cell membrane fuses with one another in such a way that they form permeable communications. Okay, so in this intercalated discs, this is not like a solid or impermeable. This is actually permeable. This intercalated discs are permeable. That allows ions to pass through and through this barrier. That means it is known as gap junctions. Okay, so intercalated discs are actually contain gap junctions. So here you can see the ions like potassium ions, sodium ions can easily pass through this intercalated discs. Okay, so we will discuss about the gap junctions and their use. Our heart is actually composed of two syncytiums. Okay, so our heart is actually composed of two syncytiums. That means it is composed of number one, atrial syncytium, and ventricular syncytium. Okay, and they are separated by, these two are separated by a fibrous ring structure that is known as fibrous annulus. That means let's try to understand this concept. If we consider that this is our atrial chamber, okay, and this is our ventricular chamber, so in between them there is a fiber separating these two. So this is called as fibrous annulus. So this is fibrous annulus. So what is the function of fibrous annulus? It is used, its work is to separate the atrial chamber from the ventricular chamber. Okay. Till now we have covered the definition of ECG, the uses of ECG and a few basic points. Okay, so to understand ECG, you must know about the electrical activity of heart. And for better understanding of the electrical activity of heart, you should first know about the electrical activity of myocardial cells. So we have to understand the electrical activity of myocardial cells. Let's draw a ventricular myocardial cell. Okay, so here is a ventricular myocardial cell. So I have drawn two ventricular myocardial cells. So let's understand the action potential generation. That means generation of action potential and a propagation of action potential across these ventricular myocardial cells. So at first, you know that the resting membrane potential of this ventricular myocardial cell is about minus 90 millivolt. So that is minus 90 millivolt. So lots of negative ions are there in this ventricular myocardial cells, and lots of positive ions are outside these cells. Now if we stimulate this myocardial cell, that means we are applying any stimulus. So after applying this stimulus, few of these positive ions gets into the cells. That means few positive ion. In case of ventricular myocardial cell, these are sodium ions. So these sodium ions are gets into the cells. So if we apply the threshold stimulus, that means if this stimulus is strong enough to generate the threshold potential, then the stimulus can also generate the action potential. So at first our cell is at rest, that is resting membrane potential of minus 90. So after applying the stimulus, the positive ions gets in and elevates this membrane potential. So let's suppose it reaches to minus 80 millivolt, then minus 70 millivolt. Okay, so as minus 70 millivolt is the threshold potential of this ventricular myocardial cells, that means now it is able to generate action potential. As I have told you earlier that at threshold potential the voltage gated sodium channels are open. So in these cells there are lots of voltage gated sodium channels. So these are voltage gated sodium channels. So after the cell reaching the threshold potential, the voltage gated sodium channels are open and lots of sodium ions get in. That means if you take the reference level like that, at first point it is here at minus 90 millivolt, then it reaches to number 2, that is threshold potential of minus 70 millivolt. Now at 70 millivolt these voltage gated sodium channels are opened. That means now the voltage, this membrane potential reaches to the 0. Okay, so that is 0 potential, minus 60, 50 millivolt. Now our cell membrane potential is 0, but at 0 potential the sodium channels are not closed, they are still open. So now this membrane potential reaches to positive value, that is plus 10 millivolt. Okay, so membrane potential is plus 10, that is opposite of minus 19. Okay, so minus, here normally it is minus, now it is positive, plus. So it is called depolarization. So when the cell reaches to plus 10 millivolt or other positive value, now the voltage gated calcium, now the calcium and potassium channels are open. In the cells there are lots of calcium channels and lots of potassium channels also. Now I have told you that the extracellular concentration of calcium is more. Okay, so extracellular calcium is more, and extracellular potassium is less. Okay, so due to this difference in concentration, at this stage potassium channels are loosed by the cells. That means these potassium channels go out. Okay, so as this potassium channel go out, the positive value of the membrane potential get decreased. So it's like that. Now when the membrane potential reaches 0, the calcium channels are also opens. So calcium channels opens. That means lots of calcium ions get into the cells. Now for a few time calcium get in and potassium go out. That means it balances the membrane potential. So it is tends to 0 for few times. Okay, now after few time these calcium channels get closed, so no extracellular calcium get in. That means it is closed, but the potassium channels are still open. So as the potassium channel are still open, the membrane potential now decreases until this reaches the resting membrane potential. Okay, so when the membrane potential reaches minus 90 millivolt value, that means the resting membrane potential, the potassium channels now get closed. Okay, so this is the process of generation of action potential. Okay, now let's have a summary. So at first we have applied stimulus. So after application of this stimulus, the sodium ions gets in. Sodium ions are positively charged, so it increases the membrane potential, which is initially as minus 90 millivolt of resting membrane potential. It increases to minus 80, then minus 80, then minus 70, and further values. So at minus 70 millivolt, which is the threshold of this ventricular myocyte, the voltage gated sodium channels are open. So due to this opening of voltage gated sodium channels, lots of extracellular sodium gets into the cells. Okay, so extracellular concentration of sodium is more and intracellular concentration of sodium is less. Due to this concentration gradient, the extracellular sodium gets into the cells and increases this membrane potential to 0 and to plus 10 or other further positive values. So at the plus 10 membrane potential, the potassium channels are opened. As I have told you, the intracellular concentration of potassium is more and extracellular concentration of potassium is less. So intercellular potassium effluxes. So due to this efflux, the membrane potential is decreased further. So when it reaches to 0, these calcium channels are also open. And I have told you that extracellular concentration of calcium is more, so lots of calcium gets in. But at this time potassium channels are not closed. So potassium ions goes out and calcium ions gets in. So due to this, the membrane potential balances to 0. But after few time, the calcium channel closes. Okay, but the potassium channels are still open. That's why potassium are still losing. So losing of this positive potassium ions decreases the resting membrane potential, and it is open until the membrane potential reaches the resting membrane potential. So that is how the action potential in a cell is generated. Now this influx of sodium reaches to this next cell. In this cell membrane there are gap junctions. Okay, and gap junctions allows this sodium ions to reach the next cell. So when the next cell is introduced this positive ions, this cycle repeats. Okay, so that means it is initially at -90. After introduction of this sodium ions, it is increasing at this +10. Here are also calcium channels and potassium channels. So this cycles goes on. Okay, so stimulation of this one cell leads to stimulation of this cell, and stimulation of this cell, here are also the third cell, and here are gap junctions also. That means sodium ions can stimulate this cell also. That's how the action potential propagates. Hope this video is helpful to you. Subscribe to my channel and spread this lecture throughout your college. Okay.